Hello class, what's up, is Raj here. So today we are discussing about the immunology paper June 2022. Okay. So first question is class, what is spleen? Spleen is the filtering blood-borne antigen. Okay. What do you mean the red pulp? Red pulp is the breakdown of defective RBC. Next question class, what is lymphoid cortex? If you're talking about the lymphoid cortex, the answer is very simple, rich in B cells. That is known as lymphoid cortex. Lymphoid paracortex rich in T cells. Okay, so these are come under in the lymphoid paracortex. Next question class, define the following. Hapten. What is the hapten? Hapten is a small molecule that cannot produce immune response alone, okay, but becomes antigenic when attached to a carrier protein. Okay, so if you're talking about this class, hapten is a small molecule that cannot produce the immune response alone first, and but becomes antigenic when attached to a carrier protein, that is known as hapten. If we're talking about the cytokinins, what do you mean by it? The cytokinins are a small signaling protein released by the immune cells that regulate immunity and inflammation. So cytokinins is a small signaling protein, okay, that to work the released by the immune cells first of all, that regulate immunity and inflammation, both come under in it, that regulate the immunity and regulate the inflammation. Next, Burosha of Fabricus. So Burosha fabricus is a lymphoid organ in a birds where the B lymphocytes are mature. First of all, so Burosha fabricus is the lymphoid organs in a birds first of all where B lymphocytes are the mature. Next, polyclonal antibodies. If you're talking about the polyclonal antibodies, the answer is very simple class. These are the antibodies produced by the different B cells clones, okay, against the different epitopes of the same antigen. So in the polyclonal antibody are the antibodies produced by the different B cell clones first of all, and after that against the different epitopes, okay, against the different epitopes come under in of the same antigen. Next question class, true false? You take it. Lymphocytes are the effector cells of adaptive immune system, that is true. Next we're talking about hematopoietic stem cells are not pluripotent cells, false. Okay, APCs directly interact with the mast cells, that is also false. NK cells do not contain TNF, that is also false. So the difference you can write down in examination, helper T cell and cytotoxic T cells. So what is the difference between them? In T cells there is known as CD4 receptor, okay, and there in cytotoxic T cell that is the CD8. It activates the other immune cells in T cells, but helper T cells, but in a cytotoxic T cells that come destroy infected cells. Okay, this is recognized by the MSC2, okay, and this is recognized by the MSC1. Okay, next is secrete cytokinins in a case of CD4, okay, in a helper T cells it's release proforin and gray enzymes that come under. After that, plasma B cells and memory B cells, what is the difference between them? Provides antibodies, okay, in plasma B cells. Memory B cells provide long-term immunity, okay. These are short-lived cells and these are long-lived cells. Active during primary response in a B cells, plasma B cells. In memory B cells, active during secondary response. Secrete immunoglobulins and do not secrete antibody immediately, okay. Next, MSC class 1 and MSC class 2 molecule difference. MSC class 1 present on all nucleated cells, first present on APCs only. Next, MSC 1 interact with the CD8 cells. MSC 2 interact with the CD4 cell. Present in endogenous antigen MSC class 1 and present in exogenous antigens in MSC class 2. Important in viral immunity in MSC class 1 and MSC class 2, important in helper T cells activation. Next, self and non-self antigens, what is the difference between them? So present naturally in a body substance, that is the self antigens, non-self antigen, foreign substance presence in it. Okay. Next, self antigens normally not attack the immune response first. Next, non-self antigens trigger immune response. Okay. And self antigens recognize as own foreign. Okay. Sorry, recognize as own in a self antigens, and non-self antigens recognize as a foreign. Okay. In self antigens maintain tolerance production. Okay. In antigen, non-self antigen causes antibody. So T cells activation. What do you mean by the T cells activation? Though, so class, the answer is very simple that T cells activations occurs when antigen presenting cells presents antigen fragments to T cells. So these are come under in the T cells activation. And what the steps including are there? APC processes antigens and present it through the MSC molecules. This has come under the steps including are there. After that, T cell receptor bind with the antigen MSC complex. Number third, co-stimulatory signal activated T cells, that also come under in it. After that, cytokines are released in it. Next, activated T cells proliferate and differentiate into the effector and memory cells. So if you're talking about the activate, the T cells proliferate and differentiate into the effector and memory cells, that also including are there. Next, we're talking about the functions. So destroy infected cells first of all. Second, activate B cells. Number third, produce cytokinins. These are the function. Next, antigen antibody precipitation. Okay. So what means the antigen antibody precipitation? So when soluble antigens combine with the specific antibody in optimal proportion, large insoluble complex form precipitated. Okay. Next process, antibody binds with the antigen specifically. Second, lattice formation occurs in it. Number third, large complexes become insoluble. Number four, visible precipitate forms. And what is the importance? So first, used in immunological tests. Number second, help detect antigen antibody reactions. Okay. Number four, classical complement pathway activation. So in this classical complement pathway activation, so the classical pathway is activated by the antigen antibody complexes mainly involving IgG or IgM. The steps are including are there: antigen antibody complex activated C1 first of all. Second, C1 activates C4 and C2. Okay. Next, C3 convertase. Okay, there is a C4B2A is formed. Okay, C3 splits into C3A and C3B. Number 5, C5 convertase forms. Okay. Number 6, membrane attack complex MAC develops. And number 7, target cell lysis occurs. Now what is the function of it? Cell destruction, opsonization and inflammation enhancement including are there. After that, these are the reactions: C4B2A plus C3B, arrow C5, okay, plus convertex, C5 convertase. Simple diagram: antigen plus antibody, C1 activation, C4 plus C2 activation, C3 convertase, C5 convertase, MAC formation and the cell lysis. These are the flow diagram are there. You can see that in a simple diagram. Structure and function of IgG. The structure is very simple class: monomeric antibodies first of all. Second, Y-shaped molecules including are there. Number third, has 2 heavy chains and 2 light chains. This also including are there. Contains Fab and Fc region. There is a 2 regions contain including are there: Fab region and Fc region. Next, functions. So neutralizes the toxins and viruses first of all. Second, activates the complement system including are there. Number third, crosses placenta. Number 4, provides passive immunity. Okay. Next, enhances phagocytosis. So these all are come under it. After the principle of ELISA and RIA. ELISA full form is class enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. So antigen antibody reaction is detected using enzyme level antibodies. So these are come under ELISA, enzyme linked immunosorbent assay. The steps are including are there: antigen coated on plate, first. Second, antibody added, number third. Enzyme linked secondary antibody added, number 4. Substrate added, number 5. Color develops indicating positive reaction. Okay. Next, uses detection of HIV, first. Number second, hormone testing, number third, disease diagnosis, उसका RIA, that is known as radio immunoassay. Okay. In radio immunoassay, the principle is very clear class, uses the radioactively labeled antigen or antibody for the detections, and the uses are very simple. Hormone estimation, detection of the very small antigen quantities, clinical diagnosis are completing in it. After that, prevention of autoimmunity. So immune system prevent the autoimmunity by the self tolerance mechanism. And what types of mechanism including are there? First, deletion of the self reactive lymphocytes, first. Second, clonal energy, number third. Regulatory T cells suppress the self reactive cells, these are come under the third. Number 4, central and peripheral tolerance come under in it. After that, what is the importance? So prevent autoimmune diseases first of all. Second, maintains the body and self recognition. After that, the cytokinins and differentiation of the CD4 plus and CD8 plus T cells. So the cytokinins are the signaling molecules that regulate the immune responses, first. Second, role of CD4 plus T cells. IL-2 stimulate the proliferation, number first. Number second, IL-4 promotes the TH2 differentiation. Number third, IFN gamma promotes the TH1 differentiation. So the role of CD4 plus T cells including are there. After that, role of CD8 plus T cells. What do you mean by the role of CD8 plus T cells? IL-2 stimulates the activation first of all. Second, the cytokinins help the cytotoxic activity and the memory formation including are there. And what does a function? Cell communication, immune regulations and activation of the lymphocytes, the function including are there. After that, short notes. Monoclonal antibodies. The monoclonal antibodies are the identical antibodies that is produced by the single B cells clone using the hybridoma technology. Okay, they are highly specific, are used in diagnosis and the cancer therapy including are there. After that, opsonization. So it is the process in which the pathogens are coated by the antibodies a complement protein to enhance the phagocytosis. So these are come under the opsonization. And after that, the DNA vaccines. So DNA vaccines contain genes encoding in antigenic proteins including are there first of all. Second, they stimulate both humoral and cell mediated immunity. So these are come under in the DNA vaccines. After that, advantages: stable, safe and long lasting immunity come under the advantage of it. After that, agglutination reaction. So agglutination reaction is the clumping particulate antigens by the antibody. In the agglutinations of the clumping of particulate antigens by the antibodies. And what is the uses of it? Blood grouping, diagnosis of diseases, these are come under in it. After that, central lymphoid organs. So the central lymphoid organ are the sites where lymphocytes mature. Okay, so in the central lymphoid organ are the sites where the lymphocytes matures. Example, bone marrow, thymus. And the function is very clear class. The lymphocyte is development, so first, and second, the immune cell maturations come under it. So thank you so much class. Please like, subscribe, share my channel how much you can. Jai Hind, Jai Bharat.